Welcome to Computational Biology. Today, we are diving into the fascinating world of sequence annotation. Imagine having the complete genetic code of an organism, but not knowing what any of it means. Sequence annotation is the essential process that solves this exact problem, turning raw DNA letters into a meaningful biological map. Let us explore how this works. To understand what sequence annotation is, imagine being handed a massive book containing 3 billion letters, but it has absolutely no spaces, no punctuation, and no chapters. That is exactly what a raw DNA sequence looks like when it first comes out of a laboratory sequencing machine. Sequence annotation is the process of reading that massive block of text and adding the spaces, highlighting the important sentences, and writing notes in the margins about what those sentences mean. It is the crucial step that makes a sequenced genome actually useful to scientists. Now that we have an intuitive grasp, let us look at the formal definition. In computational biology, sequence annotation is the multi-step process of identifying the locations of genes and all coding regions in a genome, and then determining what those specific genes do. Because genomes are so incredibly large, we cannot do this by hand. Instead, this process relies heavily on computational algorithms, massive biological databases, and statistical models to make accurate predictions about the genome. This complex process is broadly divided into two main phases. First, we have structural annotation, which involves finding the physical boundaries of genomic elements. This means locating exactly where things like start codons, stop codons, introns, and exons are positioned. Once the structure is mapped out, we move to functional annotation. This second phase takes those identified elements and assigns biological meaning to them, such as predicting the biochemical function of the resulting protein or its role in a metabolic pathway. Why does this concept matter so much in the real world? Consider when scientists sequence the genome of a new virus, like SARS-CoV-2. They desperately need to know exactly which parts of the RNA code for the spike protein so they can design an effective vaccine. Sequence annotation provides this exact map. In human medicine, annotating the genome helps us find specific mutations that cause genetic diseases by comparing a patient's genes to a healthy reference genome. Without it, modern medicine and evolutionary biology would grind to an absolute halt. To annotate a sequence automatically, computers must first find an open reading frame, commonly known as an ORF. An ORF is a continuous stretch of DNA that can potentially be translated into a functional protein without being interrupted by a stop signal. But how does a computer know if an ORF is real or just random noise? It calculates the probability that a long stretch of DNA lacks a stop codon purely by random chance. If the probability of this happening by chance is extremely low, the computer confidently annotates that region as a genuine gene. This brings us to the mathematical representation of this probability. The formula is p equals 1 minus p, all raised to the power of n. Here, capital P represents the overall probability that a sequence of codons has no stop codons, by pure chance. The lowercase p represents the probability of a single codon being a stop codon, which is typically 3 out of 64 in the standard genetic code. Finally, n represents the total number of codons in the sequence. As the length n increases, the probability p decreases exponentially, making very long ORFs incredibly strong candidates for actual biological genes. Let us see this formula in action with a concrete example. Suppose a computer program finds a continuous stretch of 100 codons without a single stop codon. We want to know the probability of this happening purely by chance. We are given n equals 100 and p equals 3 over 64. We substitute these directly into our equation. Subtracting the stop codon probability from 1 gives us 61 over 64. This represents the chance that a single codon is not a stop codon. Raising this fraction to the power of 100 accounts for every codon in the sequence. The final calculated answer is approximately 0.0082, or less than 1%. Because this probability is so incredibly low, the software will flag this region as a highly probable gene. Now that we have found the genes, we must look at their internal structure. During structural annotation, we must also find exactly where non-coding regions, called introns, are spliced out of the sequence. To do this, a computer uses a mathematical tool called a position weight matrix to score a potential splice site. This matrix assigns a specific numerical score to each nucleotide at each position, based on how frequently it appears in known real biological sequences. By evaluating these scores, the computer can determine if a sequence is a true biological signal. To find the total score for a newly discovered sequence, we use this summation formula. S total equals the sum from I equals 1 to L of S sub I of X sub I. In plain words, S total represents the final calculated score of the entire sequence. The mathematical symbol sigma means we are summing up values, starting from position 1 all the way to the sequence length L. The term S sub I of X sub I represents the specific log odds score of the nucleotide X found at position I. By summing these individual positional scores, we evaluate the sequence as a whole, rather than just looking at isolated letters. Let us apply this formula to a specific example. The scanner finds a potential 4-base splice site with the sequence G, T, A, G. We pull the corresponding log odds scores from our matrix for each position. The score for G at position 1 is 2.5, T at position 2 is 3.0, A at position 3 is 1.2, and G at position 4 is 0.8. We simply add these numbers together. The total calculated score for this potential splice site is 7.5. This positive and relatively high score indicates a very strong match, telling the software to confidently annotate this exact location as the boundary of an intron. Let us visualize everything we have discussed so far. In this diagram, you can see the complete sequence annotation process. At the top is the raw, unformatted DNA sequence. Below it, structural annotation has identified the physical boundaries, breaking the sequence into a promoter, exon 1, an intron, and exon 2. The arrows pointing downward represent functional annotation. The promoter is identified as the binding site for RNA polymerase. The exons are the actual coding regions for protein domains, while the intron is flagged as a non-coding region that will be spliced out. This is how we map the blueprint of life. To wrap up our lecture, let us review the key takeaways. Sequence annotation is the vital bridge between raw genomic data and meaningful biological knowledge. We learned that structural annotation finds physical boundaries, such as open reading frames and splice sites, while functional annotation predicts the biochemical roles of those identified genes. Finally, We saw how statistical models and probability are absolutely essential to distinguish real genes from random background noise. Mastering these concepts allows computational biologists to unlock the secrets hidden within DNA. Thank you for joining this session.